Hello. This is topic 19, the motility of life. It took me a while, honestly, to finish this topic because there's so many things I need to verify myself. Okay, so the motility of life, which means motility means how life can move around, and there's a recent paper indicate as totally, I think 18. That's my image that's showing in the first page. There are 18 different ways that a cell or an organism can gain their motility. And it's not honestly, it's not well accepted. But what's more well accepted is there's five ways, five canonical or traditional ways of motility. The first is the bacteria, bacteria flagellum, and then now acting polymerization, and also there are the last three. This three motor proteins. Three motor proteins include dynin, kinesin, and myosin. In here, actually, the kinesin doesn't really move the whole cell. And here we are mainly focusing on, we call it motility, focusing on the way that a whole cell can move around, at least move a whole cell. And there's something we will not cover in this class is that there's a lot of a molecular level motility. For example, before we talk about a lot of these gated channels, this is closed and this is open, and this is a movement. Okay, this is a change of the shape of the protein. And this is, in some way, can say motility, but in this class, in this topic, we will not cover the motility at the molecular level. And also, this thing we're going to talk about later. And this is called ATP synthesis. And ATP synthesis is a protein on the surface of the membrane. All living organisms have this protein, and this protein basically there's a it's a tunnel to allow the hydrogen pump getting out in the eukaryotic cells, getting out from the mitochondria into membrane space to the mitochondrial matrix. And in the case of the bacteria, getting in from outside the cells to inside the cells. And here, this hydrogen ion moving out, it drives the rotation. It drives the rotation of the protein, and this rotation generates ATP. Why I'm talking about this one? This is a rotary motion of a protein in the whole world of life. There are only two types of proteins can do rotary motion. And and there's another argument about this one is that you see in human life, in human civilization, I'm trying. You know the drawing, it's a car and it's a wheel. So in human civilization, in human history, the invention of the wheel is an important invention. It greatly increased our own mobility. However, in life, for some reason, there's no wheel ever invented. And for why there's some hypothesis there, maybe just a coincidence, but there's some hypothesis that, for example, if you have a high mountain, a wheel animal can get from this, and it's very hard for them getting out. Maybe the reason, we don't know, but the truth, just reality is that there's no wheel in life. So and let's further go on to talk about then. So why are we trying to say this one? Because There's only rotary motion is found in the first thing, bacteria flagellum. Flagellum itself means a long, there's a bacteria, oh not this, a very long whip. Like flagellum actually means whip, a long whip-like structure in the cells. In bacteria, it may have one or two or even more. Bacteria may have multiple flagellum in its own body. And then what's the structure of the flagellum? This bacterial cell membrane. Some bacteria has one layer cell membrane. Some bacteria have 2 layers cell membrane. And doesn't matter. All of those have similar structure. And this is called a rotor. This is a rotor of a flagellum. And then above the rotor, there's a rod, and there's a hook. This is called a hook. And then beyond the hook, there's a long flagellum going out there. And then where you have the power to drive this rotation, and what it's doing this rolling, rotate based on the rod. And what's to drive this? Here, a small molecule called the what's the name? Let me double check the name. Stator, yes. The stator. The small molecule called stator, yes. This is a molecule, stator, and there's are about 10 stators surrounding the rotor of the bacteria flagellum. And this rotator is not always sitting there. For example, this is rotor of the flagellum. Some of those stators getting here, some of those getting here, some of those far away, some of those far away, some getting here. And those are in a dynamic regulation. Basically, you can imagine those stators dynamically getting in the rotor, getting out, in the rotor, getting out. Whenever we get into the rotor, some hydrogen ion getting out from the cells. And this hydrogen ion getting out of the cells drive the rotor to rotate, have a rotation, and not drive the rotor, drive the stator to rotate. And the stator further drive the rotation of the stator. The drive of the stator give the torque to drive this rotor. And the rotor can drive, can rotate clockwise or counterclockwise. When you drive clockwise, this flagellum is behind the bacteria, and bacteria going forward. And then suddenly, out of nothing, this bacteria just every how to call it, every flagella suddenly change the rotation, and this change rotation have a switch in some of the rings. There's still being studied, but we are pretty sure there's a switch here to very very promptly change the status. So suddenly this bacteria change the shape, change the rotation direction from clockwise to counterclockwise. When you go counterclockwise, this is called swing, this is called tumble. In the tumble, they stop swimming forward, rather change direction. It is this change of direction is suddenly change direction and then swim to another direction. And this direction, if in a homogeneous, universally equal system, the bacterial swimming is totally random, like a tumble and swimming pattern. Randomly swimming everywhere, but non-stop. And but for example, there's a sugar here. a sugar here, and then in this cases, let me get a new one. Say this is bacteria. This is a bacteria swimming forward, that way, and that way, and that way. You see, it basically it didn't change any places. It's always surrounding here. However, in other cases, there's some sugar. And then in this, the bacteria going this way. And then in when there's no attractants, the bacteria only go here. But with attractant, they're going further. Because one is moving toward something that attract the bacteria, that's something it like, it suppress the tumble. It keeps swimming much faster. And then it going backwards. And the backwards much less, because if it against the gradient, concentrational gradient of the sugar, the tumble happened much faster. And then it can happen here. And then only, again, not very fast. And when it going toward the sugar, it can go very, very, very long, because the tumble are say suppressed when there is some chemicals showing a gradient of concentration from here and here. And then this bacteria is attracted to the sugar, and this is called chemotaxis. Chemotaxis is the fundamental mechanism to drive the directional motility of the bacteria. Not only the bacteria, all of those single cells are driven by chemotaxis. And even in our body, inside our body, all the cell migration is driven by chemotaxis. It's very important mechanism. So I think that's it. That's it for the bacteria. For the bacteria, bacterial flagellum. Now we talk about eukaryotic flagellum. A eukaryotic flagellum is called flagellum because, for example, this is I'm trying to draw it is a swarm. You can draw a swarm here. This is a long tail of the swarm. This is a flagellum. They both called flagellum because it's whip-like structure. However, the eukaryotic flagellum is totally different than the bacterial flagellum. It's something like this. The bacterial flagellum, it also has this is called the basal body. Bacteria has a basal body. The basal body here, there's no rotor, no stator. It's just the basal body of the flagellum. The eukaryotic flagellum is only to anchor the flagellum to the cells, to the cell membrane, and in the basal there's also other actins and other microtubules that anchor this flagellum to the cells. And then on the surface, not on the surface, about the cells, this called the filament parts. The filament parts has difference than this one. This the base body. If you look at this one here, three microtubule, 1, 2, 3, 4. 1, 2, 3, 4, 5, 6, 7, 8. It should be 9. I only draw 5, only 9. There's nine groups of triplets, the microtubule. I think we have introduced microtubule in topic 8 or something in the cells. You can go back to checklist 9 triplets, and then but in the flagellum, in the filament part, in the center there is 2 flagellum here, two microtubule here, and surrounding the central microtubule. 1, 2, 3, 4, 5, 6, 7, 8. Okay. This is called 9 plus 2 arrangement. And in between each of those, each of the 2, we call it outer. This is called outer doublets. Outer doublets. Between each of the 2 outer doublets, these are protein. The protein connect one microtubule to another, one doublet to another doublet. They call it dining. Dining is one of the motor protein, and the dining is somewhere. It has 2. Okay. Here, this dining has only. This is a motor. This is a motor, and this is a stop, and then there's a tail. The tail is anchored this way, and the motor is somewhere here, and the stop can move the other outlets in basically in this way. For example, these 2 outlets is somewhere here, okay? And then when it's moving, one is up, the other is down. So it generates a slide between these 2 outlets, and the whole thing is cool. And all 9 outlets is regularly coordinated. So in this case, it can bend the flagellum in. Not it's too much. In this called planar wave. In the planar wave, and then in the sperm, again, so the sperm tail is always in swim in this planar wave to drive them swarm forward. In this case, it's different than the couple swim pattern of this bacteria. It's not like no random turn of the eukaryotic cells. It normally smooth going forward. In the case of human sperm, the best sperm should go right forward, not left or right, only forward. But in male, that's less than 32%. So 32% of their sperm can go. If less than, let's say, 1/3 of the whole sperm cannot swim straight forward, then it will be difficult for this guy to get to have a baby. Yeah, and so this is the flagellum, and then in the this swarm is called axonal, not swarm. This dino called axonal dino, and inside the cells, there's another type of dino called cytoplasmic dino. Cytoplasmic dynan. What's the function of this dynan? Say this is a neuron, and this is long axon of a neuron. And in the long axon, here is the cell body. Here, any particles, anything that's being done here, need to be transported all the way to the terminal, axon terminal. How they can be translated? There are microtubule rail in the axon, and then this particle. What kind of thing can drive this particle? There's 2 molecules. They can drag walking along the microtubule to drag the particle, vesicle or cargo from this end to this end, or from this end to that end. And what dyning can do is that, let's say dyning is the blue one. The blue one is the microtubule, and dining will be the, say, red one. So dining, now this dining has 2 feet to drag the cargo to this side. And in the case of microtubule, this is called the minus side, and this is called the plus side. And the dining can drive the cargo from the plus side to the minus side. This is called a retrograde. Retrograde. And then another protein, let's use the green color here. Another protein also have 2 stalls and drag the cargo here. And this protein called kinesin. Kinesin can drive the cargo from the minus side to the positive side. So, and this dining and the kinesin together, they, the dining, kinesin, and this is called, I forgot, what's it? Rachel and, never mind. I cannot find it. So anyway, so these 2 molecules together mediate intracellular transportation. In dining in many cases, because it drives the flagellum of the cells. In many cases, in many protests is driven by dining. For example, Giardia. Maybe I heard Giardia is one of the #1 cause of the digestive system infection. And but let me see. Oh yeah, I think I forgot one more thing. And also in the cells. Okay, this is the let me finish this one. So this is the again just a kinning to carry cargoes from the minus side to the plus side, from the cell body to the terminal. And then the dining carry cargoes from the plus side to the minus side. And what I don't talk about the dining is that there's one more structure called cilia. Cilia is basically the same as the flagellum. The only issue is that, for example, this is a cell. This full of cilia. It's short and aloft. The cells that carry cilia on the surface, always the cell surface full of the cilia. And what's the function? Cilia mainly exist in 3 places. The one we have mentioned in previous, called ependymal cells. Ependymal cells lining the inner surface of the ventricular system of the brain and the spinal cord, and they drive or motivate or facilitate the cerebral spinal fluid to circulate inside the ventricular system. And another type of cilia cell is on the surface of respiratory system. The tract surface in our trachea or air tract surface to moving forward the mucus. Is constantly moving the mucus on the surface of the respiratory tract toward our throat to spit it out. And another thing is in the female respiratory system, more like in the uterus and in the vagina, and their function is to keep pushing out. This is so the sperm, when it get into the vagina and the uterus, they always need to moving forward against the pushing weight of the cilia. And this function of the cilia to pushing everything out is trying to help to clean up the uterus and the vagina, and also help moving the egg. The egg has no motility. So this is the cilia. And then we have talked about in human being now so far the only thing we can move the cell to move the cell to forward is the flagellum, and flagellum, and then it's only cell to have the flagellum in a human body is the sperm. And how the other cell can move? Can it move around? And the answer is yes. So the cells moving around, many cells, not all cells, but many cells have the capability of moving forward. That's moving forward, we call it cell migration. The cell migration can move forward the cells. For example, this is a cell. The cell is forming some, this is the surface. It can be an extracellular matrix or anything or something else the cell can grow on. The cell is forming adherent points on the surface on the surface, and then when the cell trying to move forward, it lose this one, and this side moving, going forward, like pushing this. This is something going forward, and this is call it laminipodia. I'm not even sure my spelling is correct. The laminipodia is all just call it podia. If it's flat, you call it laminipodia, and if it's just a point, the point normally on top of it, call it philopodia. Either way, I think I'll only test podia in this class. So, and how this happened? What this happened is that this cell surface and beneath the cell surface, there's something tiny. This is called actin fibers. Actin fibers are located beneath the cell membrane. And then if the cell, for example, disassemble the cell actin fibers here and produce more actin fibers here, and then the cells can go forward. And this is called actin polymerization. This is a relatively slow process, except for some some special cells that can move quite fast, which is the neutrophil. And most of the others are moving very slow by this way, actin polymerization. Right? And also in some other cases, let's use the blue one again. There's an actin fiber. There's another actin fiber. Let me see. Oh no, let's talk about this one first. And then when moving forward, there's another molecule. Called myosin. Myosin is another motor molecule. Now we have the third motor molecule, myosin. Myosin is not a motor molecule myosin is molecules. Yes. This is a cargo. It can carry cargo from the actin fiber also have a minus side and the plus side. It can cargo from the minus side to the plus side, like the dynesin or the kinesin. Again, kinesin carry cargo from minus side to the positive side, and dynesin carry cargo from the positive side to the minus side. And the myosin carry cargo from minus side to the positive side, to the plus side. And then this myosin can go forward carrying cargo. In some cases, this myosin, two myosin can bundle together, and even more myosin can bundle together to have a myosin bundle that has the myosin head. The myosin head also is a motor on both ends of the myosin head. And what's the benefit of this one? For example, here you can see there's two acting molecule, two acting fibers. And then if you have a myosin bundle, here is a myosin bundle. There's another myosin bundle. And then this bond, if this head can move forward, and this also can move forward, and this myosin bundle can move this two actin fiber moving closer. In the motility of the forward and mostly in the podia, the myosin mediated relative movement of the actin fiber is very critical for the motion of the actin polymerization. And furthermore, let's get a new. And furthermore here, this is a cell. And in this cell, it has many actin fibers to connect from one end to the other end. And then between each of those, there are myosins to myosin bundles to connect them. When the myosin bundles are all together, when they synchronously contract together, the cell become this, because now you have the acting closer to each other. It's hard to draw something like this. Okay. When this happens, this cells contract from larger to shorter. And what cells can contract become from longer to shorter? Muscle cells. So actin and the myosin interaction is the fundamental mechanism of the muscle contraction. And in this way, this is a smooth muscle. In a smooth muscle, the actin and the myosin bundle are not highly organized. It's more scattered around across the cells. And so this contraction is not fast or very strong, but it does do a lot of have a lot of important function in our body. For example, the stomach contraction is mainly mediated by the smooth muscle to help us mix the food. And what kind of other muscles we have? But through smooth muscle, skeletal muscle, and cardiac muscle. Cardiac muscle we'll talk about next semester. And here, next we focus on the skeletal muscle. And we can tell for sure skeletal muscle is more, stronger and faster in response compared to the smooth muscle. And how does it work? Why it work? Because in the stomach muscle, there's a special structure called sarcomere. Sarcomere is a functional unit of the muscle. It's a highly organization of the myosin bundle and the actin fibers. Let's see how they organize together. And in the very center, this is a myosin bundle. And this myosin bundle, let's say, each one of those has, let's say, I forgot, hundreds, I think 200, 300 something. And then they are so nicely, I think I should, this should be, let's draw a little bit longer. This one. they're so nicely bundled together that they have all of their heads on the surface, and actually in a rotation way, all their heads on the surface. Many of those, I only draw some of those. All their heads on the surface, and then on the two side of this bundle, not two side actually, there are actin fibers here. And then here, if you look from the microscope this way, is a kind of a little bit empty M line. This is the middle line. In this line, there's no heads here. And then all right, no, whatever. This call it Z disc. It has some shape. That's why I drawn this one. So Z disc, both actin bundles on two ends linked to a Z disc, and they are not associated together in the center. And then in these cases, when the head of the, and this is also called, and this on the microscope, you can see this one is thicker. This is called thick filaments, and this is thin filament. And then if when the heads of the myosin bind inward, the whole both group of the actin will go forward and then become closer to each other. And this is the sarcomere can contract. And this sarcomere contraction causing a muscle contraction. And how this happens? How does it happen? Because it has to be under control. How this is controlled? This is almost the last the page, I think. How this is controlled? And I cannot draw this. Whatever. This is actin fiber. The actin fiber has some site for the myosin to bind to. And once the site exposed to the myosin, you don't really have any driven power to drive the contraction of the myosin site. It automatically bind. And what's preventing the inbending is that this myosin is blocked by, this is called tropomyosin. This actin binding site, let me use some red color. This actin binding site is blocked by tropomyosin. And not really, and then the tropomyosin, let's use some other color. It's linked to the whatever it is, to the actin fiber by troponin. And then the troponin is something further. They use calcium. Calcium can bind the troponin and remove troponin from and when it bind the troponin have the conformational change. So remove the tropomyosin away from actin binding site, and then next the head of the myosin can bind to the actin to bind actin inward. That's basically how this is regulated. And yeah, so I didn't draw a lot here because it will be hard for me to draw. So, and lastly, we briefly talk about, I think this is about so far we have another topic, the muscle, which will be posted later, probably way later. Um, and we briefly talk about the motility of plants and fungi. Plants actually have some motility. It mainly made by this is the plant cell wall and cell here. This is a fluid filled vacuum, and this vacuum can change position. By changing the position, say this is a plant, and the plant can bind this word by by remove the by making the vacuum smaller on this side and longer on that side, or can bend this word by making small vacuum this side and large vacuum on the other side. And see, and then even this, there's a kind of Venus plant, I think grass. They can even capture insects as their prey by holding this together. Very powerful. And you can also watch that video. That video is fun. Some seeds can change in the how to call it, the spike outside to basically crawling on the ground. So you need to change your mind. The plants actually can moving around. They can even crawling on the ground. Some of those. But even though, however, in the major compared to animal, plants do have a reduced mobility, and some plants they can have flowers. What's the function of flowers? Attracting the insects or insects or any other animal, not really have the insects like the bats also to eat their nectar and transfer pollen for them to do the pollination for plants. So plants have more than one way of motility, or one of the ways that utilize the animal's motility. But in the case of fungi probably is the least motile species across the six kingdoms. Fungi, so far, we can say it has no way of motility. Yeast is a fungi. If you look at yeast under the microscope, it doesn't move at all. And mushroom didn't change the shape at all. Basically, but actually, what's the biggest the single organism you can find in the world? Fungi, because fungi can spread out in miles, because that's just the way of migrate. When they want to go to somewhere, they just go a long chain of fungi. What do we call it? The name, I forgot the name. They just grow. That's why they can grow so big. There's the only way they can go into a new place. They just grow and go this grow and grow. That's the way. That's their way to spread out. And again, they like they don't really have much way of motility. So that's all for today, and see you next week.